So here's something worth sitting with for a moment. Right now, as you're lying there, settling in, your body is doing about 10 million things without asking your permission. Your heart is beating. Your lungs are filling. Your cells are dividing. Your immune system is running patrol. And every single one of those processes, every chemical reaction, every electrical impulse firing across your nervous system is the product of a story that stretches back roughly 4 billion years. 4 billion. That number is so large, it basically stops meaning anything. So let's not worry too much about grasping it all at once. We've got time. We'll walk through it slowly, piece by piece, and if you drift off somewhere along the way, that's perfectly fine. Your body will keep doing all those ancient, elegant things it learned to do long before you were born. Let's start at the very beginning, and I mean the real beginning, before there was anything you'd recognize as alive. Picture Earth about 4.5 billion years ago. It's not the blue marble you've seen in photographs. It's a furious, molten, hellish place. The surface is mostly liquid rock. There's no oxygen in the atmosphere. Instead, it's thick with carbon dioxide, methane, ammonia, water vapor. The sky would look orange or reddish, perpetually hazy. Volcanoes everywhere, and the planet is getting absolutely pummeled by asteroids and debris left over from the formation of the solar system. This period is sometimes called the Hadean Eon, named after Hades, the Greek underworld, and honestly, that's a pretty fair description. It was not a hospitable place. But here's the thing. Even in all that chaos, something remarkable was happening. The planet was cooling. Slowly, over hundreds of millions of years, the surface began to solidify. Water vapor in the atmosphere started condensing and falling as rain. Rain that would have evaporated before it hit the ground at first, because the surface was still too hot. But eventually, the crust cooled enough for liquid water to accumulate. And that changes everything. Because as far as we can tell, liquid water is the essential ingredient. It's the solvent that lets complex chemistry happen. It's the medium where molecules can bump into each other, react, break apart, recombine. Without liquid water, you can have interesting chemistry, sure, but you probably can't get life. Now here's where we hit one of the biggest unsolved puzzles in all of science. How did life actually begin? How do you go from a planet covered in water and simple chemicals to something that can reproduce itself? We don't have a definitive answer, and I want to be honest about that up front, because the honesty is part of what makes this story so fascinating. We have very good hypotheses, we have experiments that show certain steps are plausible, but nobody has yet created life from scratch in a laboratory and said, "Here, this is exactly how it happened." So let's talk about what we do know and what the best thinking looks like. The classic experiment that opened this whole field was done in 1953 by Stanley Miller, a graduate student at the University of Chicago, working under Harold Urey. Miller was only 23 years old at the time, and what he did was beautifully simple. He built a closed glass apparatus, basically a loop of tubes and flasks. In one flask, he put water and heated it to create steam. The steam rose into a second flask that contained a mixture of gases meant to simulate what scientists then thought the early Earth's atmosphere looked like: methane, ammonia, hydrogen. Then he ran electrical sparks through that gas mixture to simulate lightning. The whole thing cycled continuously for about a week. When Miller analyzed the water that had collected at the bottom of the apparatus, he found amino acids. Several of them: glycine, alanine, aspartic acid. These are the building blocks of proteins, which are the molecular machines that do almost everything in your body. and Miller had made them from scratch, from simple gases and a spark. The paper he published in the journal Science in 1953 was a sensation. It suggested that the raw ingredients of life could form spontaneously under conditions that might have existed on the early Earth. You didn't need a miracle. You just needed chemistry and energy and time. Now, the Miller-Urey experiment has been debated and refined in the decades since. Our understanding of what the early atmosphere actually looked like has changed. Most scientists now think it was less rich in hydrogen and ammonia than Miller assumed, more dominated by carbon dioxide and nitrogen. But here's what's encouraging. When researchers have repeated the experiment with updated atmospheric compositions, they still get organic molecules. Maybe not as many amino acids, but they get them. And other experiments have shown that amino acids can also form in hydrothermal vents on the ocean floor, or on the surfaces of clay minerals, or even in outer space. Amino acids have been found on meteorites. A meteorite called Murchison that fell in Australia in 1969 was found to contain over 70 different amino acids. So the building blocks of life seem to be almost embarrassingly easy for the universe to produce. They show up everywhere. But amino acids aren't life. They're just ingredients. The real puzzle is how you get from a soup of organic molecules to something that can copy itself. Because that's what life fundamentally does. It reproduces. It makes more of itself, with slight variations, and those variations get tested by the environment. That's the engine of evolution, and you need it running before evolution can even begin. Most researchers today think the key molecule in the origin of life wasn't protein. It was RNA. Now, you've probably heard of DNA, the famous double helix that carries your genetic code. RNA is its close cousin. It's also a chain of nucleotide bases, but it's usually single-stranded, and it's more chemically versatile than DNA. Here's what's really clever about RNA and why it's such a compelling candidate for the first self-replicating molecule. RNA can do two things that in modern cells are handled by two completely different types of molecules. It can store genetic information, like DNA does, and it can catalyze chemical reactions like proteins do. It's a two-in-one molecule. This idea, called the RNA world hypothesis, was articulated most clearly in the 1980s, and it got a huge boost when Thomas Seck and Sidney Altman independently discovered that RNA molecules could act as enzymes. They called them ribozymes. Seck and Altman won the Nobel Prize in Chemistry in 1989 for this. Before their work, everyone assumed that only proteins could be enzymes, could speed up chemical reactions. But RNA can do it too. And that means you can imagine a very early stage of life where RNA molecules were floating around, some of them capable of copying themselves, or at least helping other RNA molecules copy themselves. No proteins needed, no DNA needed, just RNA doing double duty. Think about what that means. If you can get a self-copying RNA molecule, even a sloppy one that makes mistakes, you've got the seed of evolution. Because those mistakes, those copying errors, are mutations. Some of those mutations will make the RNA molecule better at copying itself. Some will make it worse. The ones that copy better will become more common. The ones that copy poorly will fade away. And just like that, without anyone designing anything, without any plan or intention, you've got natural selection operating at the molecular level. It's not life as we'd recognize it. There are no cells, no membranes, no metabolism in the way we usually think about it. But the fundamental logic of evolution, variation, selection, inheritance, is already running. Now, getting that first self-replicating RNA molecule to form spontaneously is still a huge challenge, and researchers like Jack Szostak at Harvard and others have spent decades working on it. The nucleotides that make up RNA are more complex than amino acids, and assembling them into functional chains without the help of existing biological machinery is genuinely difficult. But there's been real progress. In 2009, John Sutherland's group in the UK showed a plausible chemical pathway for building RNA nucleotides from very simple precursors under conditions that could have existed on the early Earth. It was a beautiful piece of chemistry, and it suggested that the problem might not be as intractable as people had feared. So let's imagine we're past that hurdle. Somehow, somewhere on the early Earth, maybe in a warm little pond, as Darwin once speculated in a letter to his friend Joseph Hooker, or maybe in the porous mineral chimneys of deep-sea hydrothermal vents, or maybe in shallow pools on volcanic islands where wet and dry cycles concentrated chemicals, somewhere RNA molecules began copying themselves, and the great experiment began. But a naked RNA molecule floating in the ocean has a problem. It's exposed, it's vulnerable to degradation, and it's competing for raw materials with every other molecule around it. What would really help is a container, a little compartment that keeps your RNA and its chemical resources together, separated from the outside world. And this is where something wonderful comes in: fatty acids. These are simple molecules with a water-loving head and a water-repelling tail, and they spontaneously form tiny spherical membranes when you put them in water. You've seen this principle if you've ever watched oil droplets in water. Fatty acid membranes don't need any biological machinery to form. They just assemble themselves, driven by basic physics. So now picture this: a self-replicating RNA molecule that happens to get enclosed inside a fatty acid vesicle. Suddenly it's got a huge advantage. Its chemical environment is more stable. The building blocks it needs for replication are concentrated inside the vesicle rather than diffusing away into the ocean. And if the RNA does something useful, like catalyzing a reaction that helps the vesicle grow, then the vesicle gets bigger, eventually splits in two, and each daughter vesicle inherits some of the RNA. That's reproduction at the cellular level. That's something you could genuinely call a protocell. Jack Szostak's lab has actually demonstrated versions of this in the laboratory. They've shown that fatty acid vesicles can grow, divide, and even compete with each other for membrane material. They've shown that RNA inside these vesicles can be copied. It's not a living cell, not yet, but it's a remarkably convincing sketch of how the first cells might have worked. And here's something to hold on to as you're lying there. The membranes of your own cells right now are made of phospholipids, which are just fancier versions of those simple fatty acids. The basic architecture, a lipid membrane enclosing a watery interior full of molecular machinery, has been conserved for nearly 4 billion years. The details have gotten spectacularly more complex, but the fundamental design is the same. You are, in a very real sense, a descendant of those first tiny bubbles of fat and RNA floating in ancient water. The oldest evidence we have for life on Earth comes from rocks in places like Greenland, Western Australia, and South Africa. In the Isua supercrustal belt in Greenland, there are rocks about 3.7 billion years old that contain carbon, with an isotopic signature suggestive of biological activity. In the Pilbara region of Western Australia, there are stromatolites, layered rock structures built by communities of microorganisms, dating to about 3.5 billion years ago. Some researchers have even claimed evidence of life in rocks from northern Quebec that might be 4.28 billion years old, though that's hotly debated. But even taking the conservative estimates, life was established on Earth by at least 3.5 billion years ago, possibly earlier. Given that the planet itself is only about 4.5 billion years old, and the first few 100 million years were that Hadean hellscape we talked about, life seems to have gotten started remarkably quickly once conditions allowed it. Some people find that reassuring. It suggests that maybe life isn't such an improbable fluke after all. Maybe, given the right conditions, it just happens. For the next billion years or so, life was entirely microbial. Single-celled organisms, bacteria and their cousins, living in the oceans, in hot springs, in shallow coastal waters, and I know that might sound boring compared to dinosaurs and whales and the drama that comes later, but this is actually where some of the most important evolutionary innovations happened. These early microbes were figuring out the fundamental chemistry of life, and the solutions they came up with are still running inside you right now. One of the biggest of those innovations was photosynthesis. And the story of how photosynthesis transformed the entire planet is one of the most dramatic events in the history of Earth, even though it was caused by organisms you'd need a microscope to see. It started with a group of bacteria called cyanobacteria, sometimes informally called blue-green algae, though they're not actually algae. What cyanobacteria figured out how to do, probably somewhere around 2.7 to 2.4 billion years ago, was use sunlight to split water molecules and harvest the energy to build organic compounds. And the waste product of that reaction, the thing they just dumped into the environment because they had no use for it, was oxygen. Now you breathe oxygen. You need it to survive. Every cell in your body depends on it. But for the microorganisms that dominated the early Earth, oxygen was poison. It's a highly reactive molecule. It rips apart the chemical bonds that early life depended on. And when cyanobacteria started pumping oxygen into the atmosphere and oceans, it triggered what's sometimes called the Great Oxidation Event, roughly 2.4 billion years ago, and it was absolutely catastrophic. Some scientists call it the oxygen catastrophe. And honestly, that name fits better. Because from the perspective of almost every living thing on the planet at that time, this was an extinction event. Maybe the first great mass extinction, and it wasn't caused by an asteroid or a supervolcano. It was caused by life itself, by the metabolic waste of trillions of tiny organisms doing nothing more than eating sunlight and exhaling. Think about that for a second. The very act of living, of being successful, of thriving and multiplying, produced a byproduct that poisoned the world. There's something almost poetic about it, in a dark sort of way. Cyanobacteria didn't mean to wreck the planet. They were just doing what life does, finding a source of energy and exploiting it. But the oxygen they released was devastating to anaerobic organisms, the ones that had been running the show for over 1 billion years. These were microbes whose entire biochemistry was built around an oxygen-free world. Their enzymes, their metabolic pathways, their cell membranes. None of it was designed to cope with this aggressive, bond-breaking molecule suddenly flooding their environment. Many of them died. Whole lineages, whole ecosystems, just gone. The survivors retreated to places where oxygen couldn't reach. Deep ocean sediments, underground aquifers, the guts of other organisms. And their descendants are still there today, by the way. Every time you hear about anaerobic bacteria in a swamp or in the mud at the bottom of a lake, those are the refugees of this ancient catastrophe, still hiding from oxygen 2.5 billion years later. But here's the thing. The great oxidation event didn't happen overnight. It wasn't like someone flipped a switch. For hundreds of millions of years before oxygen started accumulating in the atmosphere, cyanobacteria were already producing it, but the oxygen was being soaked up almost as fast as it was released. The oceans were full of dissolved iron, and oxygen reacts with iron to form iron oxides, rust, basically. So for an enormously long time, all that oxygen was just rusting the oceans, and we can actually see the evidence of this. There are these beautiful geological formations called banded iron formations, layers of iron-rich rock alternating with silica-rich layers, and they're found all over the world, in Western Australia, in the Iron Ranges of Minnesota, in South Africa, in Brazil. These bands tell a story. Each iron-rich layer represents a period when oxygen was being produced and reacting with dissolved iron, pulling it out of the water and depositing it on the ocean floor. The iron ranges that built the American steel industry, the ore that forged the ships and railroads and skyscrapers of the industrial age, that iron was laid down billions of years ago by the metabolic activity of microbes. The steel in a bridge you drive over might contain iron atoms that were rusted out of an ancient ocean by cyanobacteria during the Proterozoic. That's a connection across time that's almost hard to wrap your mind around. Eventually, though, the dissolved iron ran out. The oceans were scrubbed clean of it, and once that sink was gone, the oxygen had nowhere to go except up, into the atmosphere. That's when concentrations really started to climb, and that's when the crisis hit in full force. Atmospheric oxygen levels rose from essentially 0 to maybe a few percent, still much lower than today's 21%, but enough to fundamentally alter the chemistry of the planet. The sky changed. The oceans changed. The very rocks on the surface started weathering differently. And there's a theory, still debated but widely discussed, that this surge in oxygen triggered one of the most severe ice ages in Earth's history, the Huronian glaciation, sometimes called Snowball Earth, roughly 2.4 to 2.1 billion years ago. The idea is that oxygen reacted with methane in the atmosphere, and methane was a powerful greenhouse gas that had been helping keep the planet warm. Remove the methane, and temperatures plummet. Ice sheets may have extended all the way to the equator. The entire planet, or nearly so, locked in ice. Now, whether it was truly a full snowball or more of a slush ball, with some open water remaining near the equator, is something geologists still argue about. But the evidence for extreme glaciation at this time is strong. You find glacial deposits, called diamictites, in rocks of this age, on multiple continents, and the timing lines up suspiciously well with the rise of oxygen and the destruction of atmospheric methane. So it's plausible that cyanobacteria didn't just poison the biosphere, they also froze it. Life nearly destroyed itself through its own success. And yet, life persisted. It always does somehow. Under the ice, in hydrothermal vents, in pockets of liquid water wherever they existed, microbes hung on. And here's where the story takes one of its most important turns. Because the rise of oxygen didn't just kill things, it also opened the door to an entirely new kind of metabolism, one that would eventually make complex life possible. Aerobic respiration, using oxygen to burn fuel, is dramatically more efficient than the anaerobic pathways that came before it. When you break down glucose without oxygen, through fermentation, you get about 2 molecules of ATP, the energy currency of the cell. When you break down glucose with oxygen, you get something like 30 to 36 ATP. That's roughly an 18-fold increase in energy yield from the same amount of food. And energy is everything in biology. Energy is what lets you grow bigger, move faster, build more complex structures, maintain more sophisticated cellular machinery. Without that energy boost, complex multicellular life as we know it probably couldn't exist. You wouldn't have the metabolic budget for it. But getting from single-celled organisms to something more complex required another leap. And this one is genuinely one of the strangest and most beautiful stories in all of biology. It's the story of endosymbiosis, and it was championed by a scientist named Lynn Margulis, and the way she fought for this idea is a story worth telling. Lynn Margulis was a biologist at Boston University, and in 1967, she published a paper proposing something that most of the scientific establishment thought was, frankly, ridiculous. Her argument was that certain organelles inside eukaryotic cells, specifically mitochondria, the little structures that perform aerobic respiration, and chloroplasts, the structures in plant cells that perform photosynthesis, were not originally part of those cells at all. They were once free-living bacteria that had been engulfed by a larger cell and, instead of being digested, had survived inside their host. Over time, the relationship became permanent. The engulfed bacteria became organelles, integral parts of the host cell, and neither could live without the other anymore. This was not a brand new idea. A Russian botanist named Konstantin Mereshkovsky had proposed something similar back in 1905, and others had floated related concepts. But Margulis marshaled the evidence systematically and made the case with real rigor. She pointed out that mitochondria and chloroplasts have their own DNA, separate from the cell's nuclear DNA. That DNA is circular, like bacterial DNA, not linear like eukaryotic chromosomes. These organelles have double membranes, the inner membrane likely being the original membrane of the engulfed bacterium, the outer one from the host cell's engulfing process. They replicate by dividing, much like bacteria do. They have their own ribosomes, and those ribosomes are more similar to bacterial ribosomes than to the ribosomes in the rest of the eukaryotic cell. The evidence was compelling, but the idea was so radical that her paper was rejected by about 15 journals before it was finally published. 15. And even after publication, she faced years of skepticism and pushback. The mainstream view was that organelles had evolved gradually within cells, not that they were captured organisms. But Margulis kept pushing, kept gathering evidence, and over the following decades, molecular biology proved her right. DNA sequencing showed that mitochondrial genes are closely related to a group of bacteria called alpha proteobacteria. Chloroplast genes are closely related to cyanobacteria. Yes, the same cyanobacteria that caused the great oxidation event. So in a beautiful twist, the organisms that flooded the world with oxygen eventually became part of the cellular machinery that other organisms used to harness that oxygen. The poison became the power source, and the poisoner became a permanent passenger. This event, the engulfing of one cell by another to create a new, more complex kind of cell, is called endosymbiosis, and it's thought to have happened somewhere around 1.5 to 2 billion years ago, though the exact timing is debated. What's not debated is how important it was. This is the origin of the eukaryotic cell, the type of cell that makes up every animal, every plant, every fungus, every protist on Earth. Your cells are eukaryotic. They have a nucleus. They have mitochondria. And if you trace the lineage of those mitochondria back far enough, you arrive at a free-living bacterium that was swallowed by an archaea-like host cell in some ancient ocean, and instead of dying, it struck a deal. I'll make energy for you. You keep me safe. And that deal has held for nearly 2 billion years. Now, Margulis actually went further than mitochondria and chloroplasts. She proposed that other cellular structures like cilia and flagella also originated through endosymbiosis from engulfed spirochete bacteria. That part of her theory has not been supported by the evidence, and most biologists don't accept it. But the core insight, mitochondria and chloroplasts as endosymbionts, is now textbook biology, one of the most important ideas in the history of the life sciences. And it's worth noting that Margulis never won a Nobel Prize for it, which many people think is an oversight. She was elected to the National Academy of Sciences in 1983 and received the National Medal of Science in 1999, but the Nobel eluded her. She passed away in 2011. So now you have eukaryotic cells, cells with internal compartments, with mitochondria cranking out ATP, with a proper nucleus housing the DNA. These cells are bigger, more complex, and more energy-rich than anything that came before. And for a long time, a very long time, that's where things stayed. Single-celled eukaryotes, drifting through the oceans, evolving slowly. This period, from roughly 1.8 billion to about 800 million years ago, is sometimes called the boring billion. And the name is a little unfair, but not entirely wrong. Geologically, things were relatively stable. Oxygen levels were low but present. Life was there, but it wasn't doing anything particularly flashy by our standards. No animals, no plants, no multicellular organisms that we can clearly identify in the fossil record, at least not until near the end of that stretch. But underneath that apparent monotony, important things were happening at the genetic level. Eukaryotic cells were developing sexual reproduction, the ability to shuffle and recombine genetic material between individuals, which massively accelerates the rate at which evolution can explore new possibilities. They were developing more sophisticated ways of regulating gene expression, turning genes on and off in response to environmental signals. And somewhere in this long, quiet billion years, the first tentative experiments in multicellularity were beginning. Cells were starting to stick together, to cooperate, to specialize. Not in any dramatic way, not yet. But the groundwork was being laid for everything that would come later, for bodies, for organs, for brains, for eyes, for you. And then, around 800 million years ago, the planet shook itself awake again. Another snowball Earth event, maybe two of them in relatively quick succession. The Sturtian glaciation around 717 million years ago, and the Marinoan around 650 million years ago. Ice from pole to pole, or close to it. And when the ice finally melted, when the world warmed again, something extraordinary happened. Life exploded. Not literally, of course, but in terms of diversity and complexity. The period that followed the last snowball Earth is when things started getting really, truly interesting. Because this is when we start to see the first animals. Real, recognizable animals with bodies and symmetry and guts and mouths. And the story of how we got from single cells sticking together to actual creatures. That story is one of the most dramatic chapters in all of natural history. So let's set the scene. The Marinoan glaciation ends around 635 million years ago. The ice retreats. Meltwater pours into the oceans, carrying with it minerals and nutrients that had been locked up in glaciers. Volcanic activity during the glaciation had been pumping carbon dioxide into the atmosphere the whole time the ice was there, because volcanoes don't care about glaciers. They just keep erupting. So when the ice finally melts you've got a world with a thick greenhouse blanket, warming rapidly, and oceans suddenly flush with phosphorus and iron and all the raw materials that life needs to thrive. Oxygen levels start climbing. Not to modern levels, not yet, but significantly higher than they'd been during the Boring Billion. And that rising oxygen is crucial because oxygen is the fuel that makes complex, energy-hungry, multicellular life possible. Remember what we talked about with aerobic respiration. That roughly 18-fold increase in energy yield per molecule of glucose compared to fermentation. You need that kind of energy budget to run a body. A single cell can get by on less. But if you want muscles, if you want a nervous system, if you want to actively hunt or swim or burrow, you need oxygen. Lots of it. And this is where we meet one of the most mysterious fossil assemblages in all of paleontology, the Ediacaran biota. Named after the Ediacara Hills in South Australia, where the first really spectacular specimens were found in the 1940s and 50s by a geologist named Reg Sprigg. And here's a great little story about Sprigg. He was a young Australian geologist, and in 1946 he was doing survey work in the Flinders Ranges of South Australia, mapping old mining sites. And he found these impressions in the rock. These strange, soft-bodied forms preserved as casts and molds in sandstone. They looked biological. They looked like jellyfish, maybe, or some kind of frond-like organisms. But the rocks they were in were Precambrian, older than the Cambrian period, older than any known animal fossils at the time. And the prevailing wisdom in the 1940s was that complex life didn't exist before the Cambrian. That was basically dogma. Life before the Cambrian was supposed to be microbial, period. So Sprigg submitted a paper to Nature. And Nature rejected it. He presented his findings at a geological congress in London in 1948, and the response was polite indifference mostly. People didn't know what to make of these things. Were they really fossils? Were they just geological artifacts? Weird mineral formations that happened to look biological? Sprigg was frustrated. He eventually moved on to other work, went into the oil industry, made a successful career elsewhere. But those fossils sat there, waiting. It wasn't until the late 1950s and into the 1960s that the scientific community started taking the Ediacaran fossils seriously. A schoolboy named Roger Mason and a girl named Tina Negus independently found a spectacular specimen in Charnwood Forest in England, a frond-like fossil now called Charnia, preserved in rocks that were unambiguously Precambrian. And similar fossils started turning up in Namibia, in Russia's White Sea region, in Newfoundland, in China. This was a global phenomenon. Something was alive in the oceans before the Cambrian, something big enough to see with the naked eye, something with structure and form. But here's what's so wonderfully strange about the Ediacaran organisms. We still aren't entirely sure what most of them were. They don't fit neatly into any modern animal group. Some of them, like Dickinsonia, look like ribbed oval mats, sometimes up to a meter across, just lying flat on the seafloor. Chemical analysis of Dickinsonia fossils, and this is beautiful work done by Ilya Bobrovsky and Jochen Brox, published in 2018, found traces of cholesterol in the fossils, which is a molecule specifically associated with animals. So Dickinsonia was probably an animal of some kind. But what kind? It doesn't have a mouth. It doesn't have a gut. It doesn't seem to have had any organs at all, at least not that we can detect. It may have absorbed nutrients directly through its body surface, like a living pancake just soaking up organic matter from the microbial mats it sat on. Then you've got things like Charnia, which looks for all the world like a fern frond, but it lived in deep water, well below the reach of sunlight, so it wasn't photosynthesizing. Some researchers think it might have been absorbing dissolved organic carbon from the water. Others think it might be related to modern sea pens. The honest answer is, we're not sure. And that's part of what makes the Ediacaran so fascinating. It's like looking at an alien world, a world where life was experimenting with body plans that have no modern equivalent. Some paleontologists, most notably Adolf Seilacher, argued that many Ediacaran organisms weren't animals at all, but represented a completely separate kingdom of life. He called them the Vendobionta, that went entirely extinct, leaving no descendants. A failed experiment in multicellularity. Others disagree, seeing at least some Ediacarans as ancestors or relatives of modern animal groups. The debate is ongoing, and it's one of those wonderful scientific arguments where every new fossil discovery has the potential to shift the balance. Now think about what that means for a second. For billions of years, life on Earth was microscopic. And then, in the span of maybe 100 million years after the last snowball Earth, you get these large, complex, visible organisms appearing in the oceans around the world. Something unlocked, whether it was rising oxygen or the ecological opportunities created by the glaciations, or some genetic innovation in how cells could communicate and organize. Probably all of those things working together. Life crossed a threshold. And once it crossed that threshold, things started moving fast. Well, fast by geological standards, because the Ediacaran period, which runs from about 635 to 539 million years ago, was really just the opening act. The main event, the thing that every biology textbook talks about, is what came next. The Cambrian explosion. And the Cambrian explosion is genuinely one of the most astonishing events in the history of life. In a span of maybe 20 to 25 million years, starting around 539 million years ago, nearly all of the major animal body plans that exist today appeared in the fossil record. Arthropods, mollusks, chordates, our own group, echinoderms, annelid worms, brachiopods. Suddenly, or at least suddenly by the standards of deep time, the oceans were teeming with animals that had hard shells, jointed legs, compound eyes, guts, circulatory systems, nervous systems, predatory appendages. The jump in complexity is staggering, and the person who first really brought this to public attention, who made people understand how extraordinary the Cambrian explosion was Charles Doolittle Walcott. In 1909, Walcott, who was the secretary of the Smithsonian Institution at the time, basically the most powerful scientist in America, was riding on horseback through the Canadian Rockies in British Columbia, near a place called Field. And the story goes, though some historians question the exact details, that his horse stumbled on a piece of shale on the trail, and when Walcott looked down, he saw fossils. Extraordinary fossils. Soft-bodied organisms preserved in exquisite detail in fine-grained mudstone. He had stumbled onto what we now call the Burgess Shale, and it's one of the most important fossil sites ever discovered. The Burgess Shale is roughly 508 million years old, right in the thick of the Cambrian, and what makes it so special is the preservation. Normally, soft tissue doesn't fossilize. You get shells, you get bones, you get teeth, the hard parts. But the Burgess Shale captured everything. Eyes, gills, digestive tracts, even the contents of animals' last meals. It's like a snapshot of an entire Cambrian marine ecosystem, frozen in stone. Walcott spent years quarrying the site, pulling out tens of thousands of specimens. He tried to classify them into existing groups, shoehorning them into known categories. But decades later, in the 1960s and 70s, researchers like Harry Whittington, Derek Briggs, and Simon Conway Morris at Cambridge went back and re-examined Walcott's collections, and they realized that many of these animals were far stranger than anyone had appreciated. There was Anomalocaris, a predator up to a meter long, enormous by Cambrian standards, with circular jaws and grasping appendages at the front of its head. There was Opabenia, which had 5 eyes and a flexible proboscis extending from its face like a vacuum cleaner nozzle. When Conway Morris first presented a reconstruction of Opabenia at a conference, the audience reportedly burst out laughing because it looked so bizarre. There was Hallucigenia, which was originally reconstructed upside down and backwards. What scientists first thought were legs turned out to be spines on its back, and what they thought was the head turned out to be the tail. These animals were so alien, so unlike anything alive today, that they genuinely challenged our understanding of how evolution works. And here's the thing that really matters about the Cambrian explosion, the deep question that scientists are still wrestling with. Why then? Why, after 3 billion years of mostly microbial life, did animal body plans suddenly diversify so rapidly? There's no single answer, but there are several threads that weave together beautifully. One thread is ecological. The evolution of predation, animals eating other animals, created an evolutionary arms race. Once you have predators, suddenly there's intense selective pressure to evolve defenses. Hard shells, spines, burrowing behavior, speed, better sensory organs. And better defenses drive predators to evolve better attack strategies. It's a feedback loop, an escalation, and it drives rapid diversification. Andrew Parker, an evolutionary biologist, proposed what he called the light switch theory, the idea that the evolution of the eye, specifically the compound eye in early arthropods like trilobites, was a key trigger. Once predators could see, the whole game changed. Prey needed camouflage, or armor, or speed, or the ability to hide in sediment. Vision turned the ocean into a fundamentally different kind of environment. Another thread is genetic. The Hox genes, a family of master regulatory genes that control body plan development, that tell a developing embryo which end is the head and which is the tail, where the legs go, where the eyes form. These genes were already present in the common ancestor of all animals. They evolved before the Cambrian explosion, probably during the Ediacaran, or even earlier. So the genetic toolkit for building complex bodies was already in place. What the Cambrian explosion represents in some sense isn't the invention of new genes so much as the deployment of existing genetic tools in new combinations, in new environments, under new selective pressures. It's like the difference between inventing the transistor and building a computer. The components existed, but something catalyzed their assembly into dramatically new configurations. And then there's the environmental thread. Oxygen. This keeps coming back, doesn't it? Oxygen levels appear to have risen significantly right around the Ediacaran-Cambrian boundary. Not smoothly, not steadily, but in pulses. And each pulse of oxygenation seems to correlate with a pulse of evolutionary innovation. More oxygen means more energy available for building and maintaining complex bodies. It means you can be bigger, you can be more active, you can afford the metabolic cost of a brain or a muscular system. So you've got this perfect storm: rising oxygen, the genetic toolkit for complex bodies, the ecological pressure of predation, and possibly the ecological opportunities left behind by the extinction of many Ediacaran organisms. All of these factors converging in a relatively narrow window of geological time. And the result is the world we would recognize, at least in outline. An ocean full of animals, competing, evolving, diversifying, filling ecological niches. Now, among all those Cambrian animals, the trilobites with their crystal eyes, the armored anomalocaridids, the brachiopods cemented to the seafloor, there was one small, unassuming creature that you would have completely overlooked if you'd been diving in a Cambrian ocean. A little worm-like thing, maybe the length of your thumb, swimming with a simple fin along its back and tail. It had a notochord, a flexible rod running along its body that provided structural support. It had a nerve cord running above the notochord. It had what appeared to be simple eyes and a mouth and gill slits along its throat. Its name is Pikaia, and for a long time it was considered the earliest known chordate, the earliest member of the group that would eventually include fish, amphibians, reptiles, birds, mammals, and you. More recent discoveries, particularly a fossil called Haikuishthis from the Chengjiang fossil beds in China, dating to about 518 million years ago, push the story of early chordates even further back and give us an even clearer picture. Haikuishthis had a defined skull structure, possible vertebrae, a dorsal fin. It was tiny, maybe 2 or 3 centimeters long, utterly insignificant in the Cambrian ecosystem. Prey, not predator, the kind of thing a Anomalocaris would have eaten by the dozens without noticing. But that little creature, or something very much like it, carried within its body the basic architectural plan that would eventually produce every vertebrate that has ever lived, every shark, every salmon, every frog, every dinosaur, every sparrow, every whale, every human being. All of us trace back to something like that unassuming little swimmer in a Cambrian sea. And what happened next, over the following hundreds of millions of years, is the story of how that basic plan was modified, elaborated, tested, and transformed as life moved from the ocean onto land, and as the world above the waterline became, for the first time, a place where complex life could survive. So let's stay with those early vertebrates for a while, because what happens to them over the next couple 100 million years is one of the most extraordinary engineering stories in the history of life. And I use the word engineering deliberately, even though nobody was drawing blueprints. Evolution is, in a very real sense, a design process. It's just one that works without a designer. Through iteration after iteration, each one tested ruthlessly against the environment, and the modifications that vertebrates underwent as they diversified in the oceans and eventually hauled themselves onto dry ground. Those modifications are as elegant and as clever as anything human engineers have ever produced. Let's start in the water, because vertebrates spent a very long time there before anyone crawled onto a beach. After Haikuishthis and its kin, the next big chapter belongs to the jawless fishes. For tens of millions of years in the Ordovician and Silurian periods, roughly 485 to 420 million years ago, the most prominent vertebrates in the ocean were creatures that looked a bit like armored tadpoles. They're called ostracoderms, and they had bony plates covering their heads and sometimes much of their bodies, but they had no jaws. Their mouths were simple openings, good for filter feeding or sucking nutrients from the seafloor, but not much else. Think about what that means. You're a vertebrate. You've got this increasingly sophisticated body plan, muscles, a notochord stiffening into something like a spine, a brain protected by a skull, but you can't bite anything. You can't grab prey. You can't chew. You're limited to whatever you can vacuum up from the mud. And then, somewhere around 430 to 440 million years ago, something remarkable happened. Some lineage of these jawless fish, underwent a modification to their gill arches, the bony or cartilaginous supports that held their gill slits open. The front pair of gill arches gradually, over many generations, shifted forward and hinged, becoming the first jaws. This is one of those facts that sounds almost too strange to be true, but the evidence is really solid. The bones that form your jaw, and the jaws of every shark, every lizard, every dog, are evolutionarily derived from structures that originally supported gills. The developmental biology backs this up beautifully. In embryos, you can actually trace how the same sets of cells that form gill structures in fish contribute to jaw and ear structures in mammals. It's the same toolkit, repurposed. And here's the thing about jaws, they changed everything. Suddenly you could bite, you could grab, you could crush shells, tear flesh, grip prey. The evolution of jaws was, ecologically speaking, like the invention of the hand tool. It opened up entirely new ways of making a living. Jawed fish radiated explosively. By the Devonian period, roughly 420 to 360 million years ago, they dominated the ocean so thoroughly that the Devonian is sometimes called the Age of Fishes. And among those jawed fish, several major groups emerged that matter enormously for our story. There were the placoderms, heavily armored jawed fish, some of them absolutely enormous. Dunkleosteus, which lived about 360 million years. Ago was maybe 6 to 8 meters long, with massive bony plates on its head and jaws that could snap shut with incredible force. Some biomechanical analyses suggest its bite force rivaled that of a large alligator. It didn't have teeth in the way we think of them. Instead, those bony jaw plates were sharpened into self-shearing blades. Every time it closed its mouth, the plates slid past each other and stayed razor-sharp, like self-sharpening scissors. That's a genuinely brilliant piece of natural engineering. There were the cartilaginous fish, the ancestors of modern sharks and rays, whose skeletons were made of cartilage rather than bone. And there were the bony fish, which split into two critically important groups. The ray-finned fish, whose descendants make up the vast majority of fish species alive today, your trout, your tuna, your goldfish, and the lobe-finned fish, whose fins were thick and muscular, supported by bones that articulated in ways eerily similar to limbs. And it's that second group, the lobe-finned fish, that matters most for us right now. Because they're the ones that eventually walked out of the water. Now, let's slow down here, because the transition from water to land is one of those stories that deserves real space. It's arguably the most dramatic habitat shift in vertebrate history. Think about what's involved. In water, you're buoyant. The water supports your weight. On land, suddenly gravity is your full-time enemy, and you need a skeleton and musculature strong enough to hold you up. In water, you breathe by passing water over your gills and extracting dissolved oxygen. On land, you need lungs, or at least some way to pull oxygen directly from the air. In water, your skin stays moist automatically. On land, you'll desiccate unless you have some way to retain water. In water, your eggs can be released and fertilized externally, floating freely. On land, exposed eggs dry out and die. Your sensory systems change too. Sound travels differently in air than in water. Light behaves differently. Even the way you smell things has to be reorganized. Every single one of these problems had to be solved. And the astonishing thing is that many of them were solved before anything actually walked on land. This is a point that can't be overstated. Lungs, for instance. We tend to think of lungs as a land adaptation, but they actually evolved in fish, in water, probably as a supplementary breathing organ for fish living in shallow, oxygen-poor environments. Modern lungfish still have functional lungs and can breathe air. The swim bladder that most ray-finned fish use for buoyancy control is actually a modified lung. Evolution repurposed an air-breathing organ into a flotation device, which is almost the opposite of what you'd intuitively expect. Similarly, those muscular, bony lobe fins weren't built for walking. They were built for navigating complex underwater environments, pushing through vegetation, propping up on the bottom of shallow pools, maybe even hauling across short stretches of mud between bodies of water. But the structural elements were there, bones homologous to your humerus, your radius and ulna, even precursors to wrist and finger bones. The architecture of your arm was being roughed out in the fins of fish that had no intention of ever leaving the water permanently. The fossil record for this transition has gotten remarkably good. One of the most famous discoveries is Tiktaalik, found in 2004 by Neil Shubin, Ted Dashler, and Farish Jenkins on Ellesmere Island in the Canadian Arctic. Shubin's team went looking there deliberately. They knew from geological maps that Ellesmere had exposed rock of exactly the right age, late Devonian, about 375 million years old, and exactly the right type, the kind of freshwater sedimentary deposits where transitional fish to tetrapod fossils might be preserved. They spent several field seasons finding nothing much, and then, in 2004, there it was, Tiktaalik rosei, a creature about 2 to 3 meters long with a flat, crocodile-like head, a neck that could move independently of its shoulders. Fish don't have necks, by the way, that was new, and front fins that contained bones corresponding to a shoulder, an elbow, and a wrist. It could do a kind of push-up. It had both gills and lungs. It had scales, but also ribs robust enough to support its body in a gravity environment. It was, as Shubin has described it, a fish with a wrist. Not yet a land animal, not entirely a fish anymore. Something in between, beautifully, undeniably in between. And Tiktaalik wasn't alone. There's a whole series of these transitional forms from the late Devonian, Panderichthys, Elpistostege, Acanthostega, Ichthyostega, each showing slightly different combinations of aquatic and terrestrial features. Acanthostega, described in detail by Jenny Clack from specimens found in Greenland, is particularly fascinating because it had 8 fingers on each hand. 8, not 5. The idea that land vertebrates always had 5 digits is wrong. The earliest ones experimented with 6, 7, 8. 5 just happened to be the number that stuck, and nobody is entirely sure why. Jenny Clack, by the way, deserves a moment here. She spent decades working on these early tetrapods, often in conditions that were physically grueling. Fieldwork in East Greenland is no joke, and her research fundamentally reshaped our understanding of how vertebrates transitioned to land. Her 2002 book, Gaining Ground, became the definitive reference. She showed that many features we associate with terrestrial life evolved in animals that were still primarily aquatic. Acanthostega had legs and fingers, but also had well-developed internal gills. It probably couldn't have walked effectively on land at all. Its limbs may have been used for navigating underwater vegetation, or for moving through very shallow swamps. The legs came before the walking, which is a beautiful example of what evolutionary biologists call exaptation, a structure evolving for one purpose, and then being co-opted for another. By about 360 to 350 million years ago, in the early Carboniferous period, true terrestrial vertebrates, amphibians, were established on land, and the land they walked onto was becoming an extraordinary place. The Carboniferous, which runs from about 360 to 300 million years ago, is named for carbon, because this is the period that produced much of the world's coal deposits. And the reason it produced all that coal is that the land was covered in vast, dense, swampy forests. Enormous lycopsid trees, relatives of modern club mosses, but scaled up to 30 or 40 meters tall. Giant ferns, horsetails the size of trees. The atmosphere was rich in oxygen, possibly as high as 35% compared to today's 21%. And that oxygen-rich atmosphere had some wild consequences. Insects, which breathe through passive diffusion via tiny tubes called tracheae, could grow much larger when there was more oxygen available. So the Carboniferous had dragonflies with wingspans of 70 centimeters, nearly 2.5 feet, millipedes over 2 meters long, scorpions the size of dogs. It was a world that would feel genuinely alien to you if you could visit it. And here's a detail that connects back to something we talked about much earlier. The reason all those dead trees piled up and eventually became coal is that, for millions of years, nothing could efficiently decompose wood. Trees had evolved lignin, the tough structural polymer that makes wood rigid, but the fungi and microorganisms that break down lignin hadn't fully evolved yet. So when a tree fell, it just stayed there. Layer after layer of dead wood accumulating, getting buried, compressed, and over geological time, transformed into coal. You're literally burning ancient Carboniferous sunlight when you burn coal. The energy that powered the Industrial Revolution was photosynthesis captured 300 million years ago by trees that fell in swamps and couldn't rot. Now, for our vertebrate story, the Carboniferous is when a critical innovation appeared, the amniotic egg. See, amphibians had made it onto land, but they were still tethered to water for reproduction. Their eggs, like modern frog eggs, had to be laid in water, or at least in very moist environments. They had no shell, no internal membranes to prevent desiccation, which meant amphibians could venture onto land to feed, but they always had to return to water to breed. It's the same constraint that limits modern frogs and salamanders today. But sometime during the Carboniferous, a group of tetrapods evolved an egg with a revolutionary new design. The amniotic egg had multiple membranes, an amnion surrounding the embryo in its own private pond of fluid, an allantois for waste storage, a chorion for gas exchange, and a yolk sac for nutrition, all wrapped in a tough, sometimes calcified shell that prevented water loss. It was, in effect, a self-contained life support system, a portable pond. The embryo could develop on dry land, far from any body of water, completely protected. And the animals that laid these eggs, the amniotes, were freed from the water in a way that amphibians never were. They could colonize the interiors of continents, move into dry uplands, exploit environments that were simply off-limits to anything still dependent on aquatic reproduction. This is one of those innovations that's easy to gloss over because eggs seem so ordinary to us. But think about what that egg actually is. It's a pressurized, temperature-regulated, waste-managing, nutrient-supplying capsule that keeps a developing organism alive in a hostile environment. If an engineer designed it from scratch, you'd call it a masterpiece of bioengineering. And honestly, that's exactly what it is. Evolution stumbled onto a design solution that aerospace engineers would recognize: a sealed, self-sustaining capsule with redundant life support systems, waste management, structural integrity, and a finite fuel supply calibrated to last exactly as long as the mission requires. The amniotic egg is a spacecraft for dry land. And here's what happened next. Pretty quickly, in geological terms, the amniotes split into two great lineages, And this split is one of the most consequential forks in the entire history of life, because one branch eventually leads to you, and the other leads to dinosaurs. The division happened based on something that might sound surprisingly mundane: the number and arrangement of holes in the skull behind the eye socket. Synapsids had one opening on each side, and diapsids had two. Those openings, called temporal fenestrae, weren't decorative. They provided attachment points for jaw muscles, allowed those muscles to bulge outward during contraction, and over time enabled more powerful and more varied biting mechanics. It's a small anatomical difference that cascaded into radically different evolutionary trajectories. The synapsids, the one-hole group, are your ancestors. And here's something that surprises a lot of people. Synapsids dominated the Earth long before dinosaurs ever showed up. During the late Carboniferous, and especially the Permian period, roughly 300 to 252 million years ago, synapsids were the top terrestrial vertebrates. Early synapsids like Dimetrodon, that's the one with the big sail on its back that everyone mistakenly calls a dinosaur, were apex predators of their time. Dimetrodon was more closely related to you than to any dinosaur that ever lived. Let that sink in for a second. That weird, sail-backed reptile-looking thing from the Permian is on your side of the family tree. Over the course of the Permian, synapsids diversified spectacularly. A later group called the therapsids evolved increasingly mammal-like features: more upright posture, differentiated teeth with incisors and canines and molars instead of rows of identical pegs, and possibly even whiskers and fur. Though the fossil evidence for soft tissues is always tricky, some therapsids, particularly the cynodonts, were developing secondary palates. That's the bony shelf that separates your mouth from your nasal passages, which lets you breathe while you chew. Think about why that matters. If you can breathe and eat at the same time, you can eat more or less continuously, which supports a higher metabolic rate, which means you can potentially be warm-blooded. It's all connected. Each little anatomical tweak feeds into a larger metabolic strategy. But then came the worst day in the history of complex life on Earth. The end Permian extinction, about 252 million years ago, was the closest life ever came to being completely snuffed out. We're talking about the loss of roughly 90 to 96% of all marine species and about 70% of terrestrial vertebrate species. The cause is now fairly well understood. Massive volcanic eruptions in what's now Siberia, called the Siberian Traps, poured out lava over an area roughly the size of Western Europe. But it wasn't just the lava. The eruptions cooked through coal deposits and carbonate rocks, releasing staggering quantities of carbon dioxide and sulfur dioxide into the atmosphere. Ocean temperatures may have risen by as much as 10 degrees Celsius. The oceans became acidic, lost their dissolved oxygen, and in some areas became saturated with hydrogen sulfide, a gas that's toxic to most aerobic life. On land, the ozone layer was likely damaged, ultraviolet radiation intensified, and ecosystems collapsed in cascading waves. It took roughly 5 to 10 million years for biodiversity to recover to anything resembling pre-extinction levels. And here's where the story takes its great pivot. The synapsids, which had been the dominant land animals for tens of millions of years, were devastated. They didn't go completely extinct. Some cynodonts survived, small and burrowing and probably nocturnal, clinging on at the margins. But the ecological playing field was wiped clean, and in the aftermath, in the early Triassic, the diapsids, the two-hole skull group, began to diversify into the empty niches. Among those diapsids were the archosaurs, and from the archosaurs came two lineages that would define the Mesozoic: the crocodilian line and the dinosaur line. The earliest dinosaurs appeared around 230 to 235 million years ago, during the Middle to Late Triassic in what's now Argentina. Animals like Eoraptor and Herrerasaurus were relatively small, bipedal, and probably not particularly dominant in their ecosystems. Early dinosaurs coexisted with all sorts of other archosaurs, and it wasn't obvious at first that dinosaurs would win. But they had some key advantages: upright pillar-like leg posture that was more energy efficient for sustained locomotion, and possibly higher metabolic rates. And then another extinction event at the end of the Triassic, around 201 million years ago, cleared away many of their competitors. After that, dinosaurs exploded into every large-bodied terrestrial niche and held them for over 130 million years. Now, while the dinosaurs were busy being spectacular, and they were, from the long-necked sauropods that were the largest land animals ever to live, to the theropods that included some of the most efficient predators evolution has ever produced. While all that was happening, your ancestors were doing something much less glamorous, but arguably just as important. They were staying small. They were hiding. And they were getting very, very good at it. The mammals of the Mesozoic are one of the most underappreciated chapters in evolutionary history. For a long time, the standard story was that early mammals were tiny, shrew-like creatures that cowered in the shadows of dinosaurs for 160 million years, just waiting for their chance. And while there's some truth to the small and nocturnal part, recent fossil discoveries have shown that Mesozoic mammals were far more diverse and interesting than anyone suspected. There were mammals that glided like flying squirrels, Velaticotherium, from about 164 million years ago in China. There were mammals that swam, like Castorocauda, a beaver-like creature from the Middle Jurassic. There was even Repenomamus, a badger-sized mammal from the early Cretaceous that had baby dinosaur bones in its stomach. So it wasn't all cowering. But the key innovations were happening at a subtler level. Mammals were refining the warm-blooded, high-metabolism strategy that their theropsid ancestors had started. They evolved true fur for insulation. They evolved mammary glands. And that's such a strange, wonderful innovation, feeding your young with secretions from modified sweat glands. It sounds almost absurd when you describe it that way, but it's extraordinarily effective. It means the mother can convert whatever food she finds into a perfectly calibrated, nutrient-rich, antibody-laden liquid food for her offspring. The baby doesn't need to be able to find or process food on its own. It just needs to find its mother. That's a massive survival advantage, especially for small animals in a dangerous world. And then there's the brain. Mammalian brains, relative to body size, were already larger than those of most contemporary reptiles. The neocortex, that wrinkled outer layer that handles sensory processing, spatial reasoning, and eventually, in our lineage, language and abstract thought, was expanding. Some researchers think the nocturnal lifestyle actually drove brain evolution in early mammals. If you're active at night, you can't rely on vision as much. You need better hearing, better smell, better tactile sensitivity through those whiskers. Processing all that sensory information requires more neural real estate. So the darkness, in a way, may have been building the hardware that would eventually let mammals think their way to dominance. Here's another thing that was happening during the Mesozoic that's easy to miss. The continents were moving. Pangaea, that supercontinent that had assembled during the late Paleozoic, was breaking apart. The Atlantic Ocean was opening up. India was drifting northward. And as continents separated, populations of animals became isolated from each other, which is one of the most powerful drivers of speciation. The mammals on what would become South America evolved independently from those on what would become Africa, which evolved independently from those on what would become Australia. This continental fragmentation set the stage for the extraordinary diversity of mammalian forms that would eventually emerge. And meanwhile, in one particular lineage of theropod dinosaurs, something remarkable was happening. Feathers were evolving. Not for flight at first. The earliest feathered dinosaurs used them for insulation, for display, maybe for brooding eggs. But over millions of years, in the Maniraptoran theropods, feathers became more complex, forelimbs became longer, and body size shrank. The boundary between small feathered dinosaur and bird is genuinely blurry. Archaeopteryx, from about 150 million years ago in what's now Bavaria, was long considered the first bird, but we now know it was just one of many small, feathered, possibly flight-capable dinosaurs. The real origin of powered flight in this lineage probably happened multiple times, in multiple closely related groups, during the late Jurassic and early Cretaceous. So by the late Cretaceous, say 70 million years ago, you've got this world that's warm, that has no polar ice caps, that's dominated by dinosaurs on land and marine reptiles in the sea and pterosaurs in the air, with mammals still mostly small but quietly diversifying, and birds already established as a successful group. And everything is about to change because somewhere out in the solar system, a rock about 10 to 12 kilometers across is on a trajectory that will intersect with Earth at a point that happens to be the shallow seas of what's now the Yucatan Peninsula in Mexico, the Chicxulub impactor. And when it arrives, 66 million years ago, it will release energy equivalent to roughly 10 billion Hiroshima bombs, punch a crater nearly 180 kilometers wide, and trigger a cascade of environmental catastrophes, superheated ejecta raining back through the atmosphere, global wildfires, a years-long impact, winter as dust and soot, block sunlight, acid rain, collapse of photosynthesis-based food chains in both the oceans and on land. Every non-avian dinosaur dies. Every pterosaur dies. The great marine reptiles, the mosasaurs, the plesiosaurs, gone. Roughly 75% of all species on Earth are wiped out in what might have been a matter of months to years for the initial kill mechanisms, followed by decades of ecological collapse. But some things survive. Crocodilians in freshwater environments, turtles, sharks, small birds, the ancestors of every bird alive today. And mammals. Small, burrowing, seed-eating, insect-eating mammals that could hunker down, that didn't need much food, that could wait out the darkness. And when the skies finally cleared, when the sunlight returned and the plants began to grow back, those survivors looked out on a world emptied of its dominant players. Every large-body ecological niche was vacant, and what happened next was one of the most explosive radiations in evolutionary history. Mammals went from small, marginal creatures to the dominant large animals on every continent in a span of just a few million years. Geologically speaking, that's a blink, and the variety of forms they produced is staggering, from tiny shrew-like insectivores to whale-sized ocean swimmers, from bats fluttering through the night sky to massive herbivores shaking the ground with every step. This period, the early Cenozoic, is sometimes called the Age of Mammals, and it earns that title almost immediately. Now, there's a term paleontologists use for this kind of rapid diversification: adaptive radiation, and it's worth sitting with for a moment, because it's one of the most beautiful patterns in all of evolution. The idea is simple. When a group of organisms suddenly finds itself with access to a huge range of empty ecological niches, ways of making a living, essentially, evolution can move fast. Not fast in the way we normally think of fast, but fast on geological timescales. New body plans, new diets, new locomotion strategies, new sensory systems, all of it blooming outward like branches on a tree, each lineage specializing in a slightly different direction. And that's exactly what mammals did. Within about 10 million years of the Chicxulub impact, you've got the ancestors of most of the major mammalian orders we recognize today. Primates, rodents, carnivores, ungulates, bats, all of them tracing their origins to this narrow window of post-extinction opportunity. And here's what's really fascinating about this. The mammals that survived the extinction weren't specialists. They were generalists. Small, flexible, adaptable creatures that could eat a range of foods and tolerate a range of conditions. That's what got them through the crisis. But once the crisis was over, the evolutionary pressure flipped. Now the advantage went to specialists, to lineages that could carve out a specific niche and exploit it better than anyone else. So you get this incredible flowering of diversity from a relatively small pool of survivors. Let's talk about some of the directions that flowering took, because they're genuinely wild. Take whales, for instance. You're lying there, maybe thinking about blue whales, the largest animals that have ever lived on Earth, larger than any dinosaur, by the way. And it might surprise you to learn that their ancestors were small, four-legged, hoofed mammals that lived on land. We know this because we have the fossils to prove it. In Pakistan and India, paleontologists have found a remarkable sequence of transitional forms. There's Pakicetus, from about 53 million years ago, a wolf-sized animal with hooves living near water, probably wading in to catch fish. Then Ambulocetus, a few million years later, bigger, more aquatic, with large feet that probably worked like paddles. Then Rhodhocetus, with shorter legs and a more streamlined body. Then Basilosaurus, which despite its name, it means king lizard, because the people who first found it thought it was a reptile, was a fully aquatic mammal, maybe 15 to 18 meters long, with tiny vestigial hind limbs that served no locomotor function at all. This whole transition from land to sea took maybe 15 million years. The skeletal evidence is so complete, so beautifully graded, that whale evolution has become one of the textbook examples of macroevolution captured in the fossil record. And think about what had to change. Not just the limbs, though watching forelimbs become flippers and hind limbs shrink to nothing is remarkable enough. The nostrils had to migrate from the front of the skull to the top, becoming a blowhole. The tail had to develop horizontal flukes for propulsion, the opposite of fishtails, which are vertical. The kidneys had to adapt to process salt water. The ears had to restructure entirely for underwater hearing. And whale ears are extraordinary, by the way, capable of detecting sounds across enormous distances through water. Every one of these changes required coordinated shifts in multiple genes, multiple developmental pathways. And yet it happened, step by step, each generation slightly better suited to an aquatic life than the last. Meanwhile, on land, another remarkable story was unfolding. The story of horses. And this one is interesting because it's one of the earliest evolutionary sequences that paleontologists ever worked out, going all the way back to the 1870s when Thomas Huxley and Othniel Charles Marsh pieced together the fossil record of horse evolution in North America. The earliest horse ancestor we know of is Hyracotherium, sometimes called Eohippus, the dawn horse, from about 55 million years ago. And if you saw one, you would not recognize it as a horse. It was about the size of a fox terrier, maybe 30 centimeters tall at the shoulder. It had 4 toes on its front feet and 3 on its back feet. It lived in forests and browsed on soft leaves. Over the next 50 million years, as grasslands spread across North America and other continents, driven partly by climate change, partly by the evolution of grasses themselves, horse ancestors got bigger, their legs got longer, their side toes shrank, and their teeth changed dramatically. Teeth are actually a huge part of this story. Grass is tough, full of silica, and it wears teeth down fast. So horse teeth became taller, with more complex ridging patterns, better suited for grinding. By the time you get to modern equus, appearing around 4 to 5 million years ago, you've got a single-toed, long-legged open grassland runner with teeth built for a lifetime of chewing abrasive grasses. It's a beautiful example of how environment shapes form over deep time. But here's the thing, and this is important. Horse evolution wasn't a straight line. Early paleontologists sometimes presented it that way, as this neat ladder from small forest browser to large plains runner. But the reality was much bushier than that. There were dozens of horse species at various times, many of them coexisting, many of them going extinct. Evolution isn't a ladder. It's a bush with many branches, most of which are dead ends. The lineage that led to modern horses is just one surviving twig on a very bushy shrub. Now, I want to come back to something I mentioned earlier, primates, because of course, that's our story. And it starts in the trees. The earliest primates appear in the fossil record around 55 to 60 million years ago, right in that post-extinction radiation window. They were small, probably nocturnal, living in tropical forests, eating insects and fruit. And from the very beginning, primates had a few distinctive features that would turn out to matter enormously down the line. Forward-facing eyes, giving them stereoscopic vision, depth perception, grasping hands with opposable thumbs, nails instead of claws, relatively large brains for their body size. All of these features are thought to be adaptations for life in the trees, for an arboreal lifestyle where you need to judge distances accurately, grab branches securely, and navigate a complex three-dimensional environment. And for tens of millions of years, primates stayed in the trees, diversifying into many lineages. The prosimians, lemurs, lorises, tarsiers, split off early. Then the anthropoids emerged, the group that includes monkeys, apes, and us. By about 35 million years ago, you've got early monkeys in Africa. By about 25 million years ago, the first apes appear, the superfamily Hominoidea. And apes were different from monkeys in some key ways. No tails, more flexible shoulders, better suited for hanging and swinging, larger brains relative to body size, broader chests. These early apes were still forest dwellers, still arboreal, and for a long time they were incredibly successful. During the Miocene epoch, roughly 23 to 5 million years ago, apes were diverse and widespread across Africa, Europe, and Asia. There were dozens of species. This was the golden age of apes. But then something shifted. The climate cooled, forests shrank, grasslands expanded, and most of those ape species went extinct, outcompeted in many environments by monkeys, which were smaller, faster reproducing, and better adapted to the changing forests. The apes that survived were pushed into refugia, shrinking patches of forest in Africa and Southeast Asia. And one lineage of African apes, somewhere between about 7 and 6 million years ago, split into two branches. One branch led to chimpanzees and bonobos. The other branch led to us. And this is where the story gets personal, doesn't it? Because now we're talking about the hominins, the group that includes modern humans and all of our ancestors and close relatives after the split from the lineage leading to chimps. And the first thing to understand is that this wasn't a single species marching steadily toward humanity. It was, again, a bush. Multiple species, often coexisting, experimenting with different ways of being almost human. The earliest hominins we know of are fragmentary and debated. There's Sahelanthropus chadensis from Chad, roughly 7 million years old, known mainly from a skull that has a puzzling mix of ape-like and hominin-like features, including a foramen magnum. That's the hole where the spinal cord exits the skull, positioned in a way that suggests it might have walked upright, at least some of the time. Then there's Ardipithecus from Ethiopia, around 4.5 million years ago. Ardi, as she's known, a partial skeleton showing an animal that could walk bipedally on the ground, but still had a grasping big toe for climbing trees. She lived in a wooded environment, not open savanna, which challenged the old idea that bipedalism evolved specifically as an adaptation to grasslands. And that question, why did our ancestors start walking upright, is one of the most debated in all of paleoanthropology. Dozens of hypotheses have been proposed: freeing the hands to carry food or tools, reducing sun exposure in hot open environments, seeing over tall grass, moving more efficiently over long distances. The truth is probably some combination, and it probably varied over time and between species. But whatever the reason, bipedalism was the first great hominin innovation, appearing millions of years before big brains, millions of years before stone tools, millions of years before language. Our ancestors walked upright long before they were smart, and that walking, that shift in posture and locomotion, reshaped the entire skeleton. The pelvis widened and tilted, the spine developed its characteristic S-curve, the legs lengthened, the feet lost their grasping ability, and became platforms for striding. Every step you take is the legacy of that ancient restructuring. By about 4 million years ago, we get the Australopithecines, and this is where the fossil record really starts to fill in. The most famous of all is Lucy, an Australopithecus afarensis skeleton found in Ethiopia in 1974 by Donald Johansen and Tom Gray. She's about 3.2 million years old, stands maybe a meter tall, has a brain about the size of a chimpanzee's, but walks fully upright. The team that found her reportedly played Lucy in the Sky with Diamonds in camp that night, and the name stuck. She became one of the most important fossils ever discovered because she showed so clearly that bipedalism came first and big brains came later. That was a crucial insight. For decades, scientists had assumed it was the other way around, that our ancestors got smart first and then stood up. Lucy proved them wrong, and the Australopithecines were diverse. There were gracile forms like Lucy's species and robust forms, Paranthropus, with massive jaws and huge chewing muscles anchored to bony crests on top of their skulls, specialized for eating tough, hard foods. Some of these robust species survived until less than 1 million years ago, coexisting with early members of our own genus. But it's from the gracile Australopithecines, or something very close to them, that the genus Homo eventually emerged, around 2.5 to 3 million years ago in East Africa, during a time of increasing climate variability, alternating wet and dry periods that may have put a premium on flexibility, on the ability to adapt to changing conditions, rather than specializing for any single environment. And with the emergence of Homo came two things that would change everything. The first is stone tools. The second is a dramatic expansion of the brain. And these two developments are deeply intertwined, feeding into each other in ways that are still being untangled today. Let's start with the tools. The oldest stone tools we know of come from a site called Lomekwi, on the western shore of Lake Turkana in Kenya. They're about 3.3 million years old, which actually predates the earliest confirmed members of genus Homo. That's a fascinating wrinkle. It means Australopithecines, or something very close to them, were already bashing rocks together to create sharp edges before our genus even existed. These Lomekwian tools are crude, heavy, not terribly refined. But they represent something extraordinary, the deliberate modification of one natural object using another to produce a tool with a specific function. That's not nothing. That's a cognitive leap. Then, around 2.6 million years ago, you get the Oldowan toolkit, named after Olduvai Gorge in Tanzania, where Louis and Mary Leakey did so much of their pioneering work. Oldowan tools are simpler than what would come later, mostly cobbles with a few flakes knocked off to create a cutting edge, but they're remarkably effective. You can butcher an animal carcass with an Oldowan flake. You can crack bones open to get at the marrow inside. And that matters enormously, because marrow is incredibly calorie-dense, rich in fat, and it was a food source that most other animals on the savanna couldn't easily access. Think about that. Here's a small-bodied, relatively weak hominin competing with hyenas and big cats and vultures for access to carcasses. And the stone tool is the great equalizer. You don't need massive jaws or powerful claws if you've got a sharp rock and the know-how to use it. And here's where the feedback loop begins. Better access to high-quality food, meat, marrow, eventually cooked food, though cooking comes much later, provides the calories needed to support a larger brain. Brains are metabolically expensive. Your brain right now, as you lie there, accounts for roughly 2% of your body weight, but consumes about 20% of your resting energy. That's an enormous cost. You can only afford a brain that big if you're taking in enough calories to fuel it, and if the payoff in terms of survival and reproduction justifies the expense. For early Homo, stone tools opened up a dietary niche that made larger brains viable, and larger brains, in turn, enabled more sophisticated tool-making, better planning, more complex social coordination. Each advance made the next one possible. The species most associated with this early toolkit is Homo habilis, handyman, who shows up in the fossil record around 2.4 million years ago. Habilis had a brain roughly 50% larger than an Australopithecines, maybe around 600 to 700 cubic centimeters compared to Lucy's 400 or so. Still small by our standards, your brain is about 1,350 cubic centimeters, but a significant jump. And habilis wasn't alone. There's Homo rudolfensis, known from a beautifully preserved skull found at Kubifora, also in Kenya, with a somewhat larger brain and a flatter face. The exact relationships between these early Homo species are genuinely messy. The fossil record for this period is fragmentary, and paleoanthropologists argue vigorously about which specimens belong to which species, or whether some of these species are really distinct at all. That's honest science. The evidence is incomplete, and the boundaries between closely related species are inherently fuzzy, especially when you're looking at populations that were actively evolving. But the broad picture is clear. Between about 2.5 and 1.5 million years ago, in East and possibly South Africa, you have multiple hominin species coexisting, some robust Australopithecines still crunching away on tough plant foods, and several early Homo species increasingly reliant on tools, meat, and social cooperation. It's not a single line marching toward us. It's a bush with multiple branches, most of which eventually go extinct. Now, around 1.9 million years ago, something remarkable happens. A new species appears that looks, for the first time, genuinely modern in its body proportions. Homo erectus. And this is a big deal. Erectus is tall. Some individuals stood nearly 6 feet. Their legs are long relative to their arms, their shoulders are broad, their body is built for walking and running long distances in open landscapes. Their brain is significantly larger again, starting around 600 cubic centimeters in the earliest specimens and eventually reaching 1,100 or more in later populations. And here's what's really striking about erectus. They're the first hominin to leave Africa. Within a few 100,000 years of appearing in the fossil record, Homo erectus populations show up in the Republic of Georgia, at a site called Dmanisi, where remarkably complete skulls dating to about 1.8 million years ago were found, and in Southeast Asia, on the island of Java, where Eugène Dubois first discovered what he called Pithecanthropus erectus back in 1891, one of the very first hominin fossils ever recognized. Dubois was a Dutch anatomist who literally quit his university job and moved to the Dutch East Indies specifically to find the missing link between apes and humans. Most people thought he was crazy. He found it. The specimen he unearthed along the Solo River, a skullcap and a femur, proved that an upright walking large-brained creature had existed in deep antiquity. It was one of the great vindications in the history of science, though Dubois spent much of his later life embroiled in bitter disputes about what his fossil actually was. Homo erectus also brought a major technological innovation, the Acheulean hand axe. If you've ever seen one, you know how striking they are. Teardrop-shaped, symmetrical, carefully flaked on both sides to create a tool that fits beautifully in the hand. The oldest Acheulean tools date to about 1.76 million years ago, from a site in Kenya, and they persist in the archaeological record for over 1 million years, making them the longest used tool design in all of human history. Think about that. We've had smartphones for about 15 years. The Acheulean hand axe was the dominant technology for 1 million years. There's something both humbling and fascinating about that kind of stability. It suggests a species that found something that worked and stuck with it, generation after generation, across continents. And erectus may have controlled fire. The evidence here is debated. Firm, unambiguous evidence for controlled fire use doesn't appear until maybe 400,000 years ago or so, at sites in Europe and the Middle East associated with later species. But there are tantalizing hints of much earlier fire use, including burned bones and sediments at Wonderwork Cave in South Africa, dating to about 1 million years ago, and at Gesher Beno Yaakov in Israel around 790,000 years ago. If erectus did master fire, even sporadically, the implications are enormous. Fire means cooking, and cooking transforms food. It breaks down tough plant fibers, denatures proteins, kills parasites, and dramatically increases the number of calories your gut can extract from a meal. Richard Wrangham, a primatologist at Harvard, has argued that cooking was the key innovation that allowed our ancestors' brains to grow so large that you literally cannot fuel a human-sized brain on raw food alone without spending most of your day chewing, the way chimpanzees do. It's a compelling idea, though the timing remains uncertain. What's not uncertain is that something was driving brain expansion, and it was accelerating. From Homo erectus onward, brain size trends upward across multiple hominin lineages. By around 600,000 to 300,000 years ago, you get species with brains approaching or matching modern human size. In Europe, this lineage becomes the Neanderthals, Homo neanderthalensis, who are far more sophisticated than the brutish caricature that persisted for most of the 20th century. Neanderthals had brains as large as ours, sometimes larger. They made complex tools, used fire routinely, buried their dead, at least sometimes. And there's evidence they made simple ornaments, used pigments, and may have had some form of symbolic expression. They were adapted to cold climates, with stocky, muscular builds and broad noses suited to warming, frigid air. They hunted large game, bison, horses, mammoths, at close range with thrusting spears, which took extraordinary courage and coordination. Their bones show frequent healed fractures, the kind of injury pattern you see in rodeo riders. They lived hard lives. In Africa, meanwhile, a parallel lineage was evolving. And this is where we come in. The oldest fossils attributed to Homo sapiens, our species, come from a site called Jebel Irhoud in Morocco, and they're about 300,000 years old. When those dates were published in 2017, they pushed back the origin of our species by about 100,000 years and shifted the geography too. Previously, East Africa had been considered the cradle of sapiens. Now it seems our species may have emerged across a broad swath of the African continent, with populations connected by intermittent gene flow as climates shifted and habitats expanded and contracted. It's a more complex picture than a single Garden of Eden, and in some ways, a more interesting one. But what made sapiens different? Physically, the answer is subtle. We have a distinctive skull shape, a high, rounded brain case, a flat face, a prominent chin. Our skeletons are notably gracile compared to Neanderthals or Erectus, lighter boned, less muscular. But the real difference, the one that would eventually matter more than anything else, seems to have been behavioral. Somewhere between 300,000 and about 50,000 years ago, something shifted in how our ancestors thought, communicated, and organized themselves. And the evidence for that shift is written in the archaeological record in a way that gets more and more vivid the closer you look. Around 100,000 years ago, maybe a little earlier, you start finding things at African sites that don't make practical sense. Shells with holes drilled through them, clearly strung as beads. Lumps of ochre, iron-rich pigment, ground into powder and sometimes etched with geometric patterns. At Blombos Cave in South Africa, dating to roughly 75,000 to 100,000 years ago, archaeologists found pieces of ochre scored with cross-hatched lines, deliberate, repeated, unmistakably intentional marks, not tool-sharpening grooves, not accidental scratches. Someone sat down and carved a pattern because the pattern itself meant something. Think about what that implies. A mind that can hold an abstract symbol, a mark that stands for something other than itself, is a mind that has crossed a threshold. It's the same cognitive leap that eventually gives you language, mathematics, art, religion, and every complex institution humans have ever built. And it didn't happen overnight. The evidence suggests it accumulated gradually, in fits and starts, across tens of thousands of years. At some sites, you find beads, then nothing for millennia, then beads again. The capacity was there, flickering, long before it became the roaring fire we associate with fully modern human behavior. By around 70,000 to 50,000 years ago, though, something accelerated. Some researchers call it the Upper Paleolithic Revolution, though that term is a bit misleading because much of the action happened in Africa during the Middle Stone Age, well before the European Upper Paleolithic. Others talk about behavioral modernity as a package, a suite of traits that include symbolic art, complex tools made from multiple components, long-distance trade networks, systematic use of fire, fishing, bone and antler working, and burial of the dead with grave goods. You don't need all of these at once to qualify, but when you see them clustering together, you're looking at minds that operate the way ours do. Minds that plan far into the future, that communicate nuanced ideas, that build social structures held together by shared stories and shared symbols. And here's what's really fascinating. Language almost certainly played the central role in this transformation, but language doesn't fossilize. You can't dig up a conversation. So we're left reading the indirect evidence, the artifacts that only make sense if the people who made them could talk to each other in sophisticated ways. Teaching someone to make a composite tool, say a spear with a stone point hafted onto a wooden shaft using plant resin and sinew bindings. That's not something you can learn just by watching. You need instruction. You need words, or at the very least, a communication system rich enough to convey sequences, conditions, and corrections. Heat the resin until it's tacky but not liquid. Wrap the sinew this way, not that way. Tighter. Now let it cool. The tools themselves are evidence of language, even though no word survives. It was around this time, roughly 70,000 years ago, give or take, that small groups of Homo sapiens began leaving Africa in a sustained way. There had been earlier dispersals, traces of sapiens in the Levant, as far back as 180,000 years ago. But those earlier waves seem to have fizzled out or remained local. The later migration, or more likely migrations, plural, took hold. People moved along the southern coast of Asia, reached Southeast Asia, and astonishingly, crossed open water to reach Australia by at least 65,000 years ago. That's a sea crossing. Even during glacial periods when sea levels were lower and more land was exposed, getting to Australia required navigating stretches of open ocean, maybe 60 or 70 kilometers at the shortest points. You need boats. You need planning. You need the ability to look at the horizon and believe there's something worth reaching on the other side. That's not just intelligence. That's imagination. And everywhere sapiens went, they encountered other human species. In Europe and Western Asia, they met Neanderthals. In parts of Eastern Asia and Oceania, they encountered Denisovans, a group we know mostly from DNA and a handful of fossils from a single cave in Siberia, though their genetic legacy is widespread in modern populations across Asia and the Pacific. And here's where the story gets beautifully complicated, because these encounters weren't just competition. They were also, at least sometimes, intimate. We know this because Neanderthal DNA makes up roughly 1 to 4% of the genome of most people alive today, whose ancestry is primarily non-African. Denisovan DNA shows up in Melanesian and Aboriginal Australian populations at even higher percentages, sometimes around 4 to 6%. These aren't trace amounts. These represent sustained interbreeding over thousands of years, enough for natural selection to act on the borrowed genes. And some of those borrowed genes turned out to be genuinely useful. Tibetan populations carry a variant of the EPAS1 gene, a gene involved in the body's response to low oxygen levels at high altitude, that appears to have come from Denisovans. It's one of the clearest examples of adaptive introgression, where a gene from another species provides a survival advantage in a specific environment. Neanderthal-derived genes in modern humans have been linked to immune function, skin and hair characteristics, and even, intriguingly, to certain aspects of sleep and mood regulation. We carry pieces of them. They're gone as a distinct population. The last Neanderthals disappearing from southern Iberia and possibly Gibraltar around 40,000 years ago, maybe a few thousand years later in some refugia. But they're not entirely gone. They live on in our cells, in the molecular machinery that helps your immune system recognize a pathogen or helps your skin respond to ultraviolet light. So why did sapiens persist while Neanderthals and Denisovans didn't? This is one of the biggest questions in paleoanthropology, and the honest answer is that we don't fully know. It probably wasn't one thing. Neanderthals were smart. They were tough. They survived ice ages that would have killed most species. But sapiens may have had advantages in social organization. Larger, more interconnected groups, more extensive trade networks, more flexible diets. There's evidence that sapiens populations maintained contact over hundreds of kilometers, exchanging raw materials and probably ideas. Neanderthal groups seem to have been smaller and more isolated. When times got hard, and the climate between 50,000 and 30,000 years ago was brutal, swinging between cold snaps and brief warm spells, that isolation may have been fatal. Small, disconnected populations are vulnerable. They lose genetic diversity. They can't bounce back from local disasters. One by one, the lights went out. By around 40,000 years ago, sapiens were the last humans standing. And then something extraordinary happened, or rather, kept happening, with increasing intensity. The archaeological record from this period, the European Upper Paleolithic, the Late Stone Age in Africa, explodes with creativity. The cave paintings at Chauvet in southern France, dating to about 36,000 years ago, are breathtaking. Lions, horses, rhinoceroses, rendered with a sophistication and dynamism that wouldn't be matched for tens of thousands of years. These aren't crude scratches. They use shading, perspective, even a kind of animation, overlapping images of legs to suggest movement. Whoever painted them understood how to see, how to represent what they saw, and how to make you feel something when you looked at it. The oldest known figurative sculpture, the Lion Man of Hohenstein-Stadel in Germany, is about 40,000 years old. A figure with a human body and a lion's head, carved from mammoth ivory with stone tools. That's not just art. That's mythology. That's a mind that can blend categories, imagine things that don't exist, and give them form. And this same creative explosion was happening in music. Bone flutes from caves in Germany, also around 40,000 years old, are the earliest known musical instruments. They're made from bird bones and mammoth ivory, with carefully spaced finger holes that produce recognizable scales. Someone sat in a cave by firelight and played music, maybe to entertain, maybe to bond a group together, maybe as part of a ritual. We don't know. But the impulse, the need to make organized sound, to create something beautiful that exists only in time, that impulse is as human as anything we do. From this point forward, the pace of change accelerated in a way that had no precedent in the history of life on Earth. For billions of years, evolution had been driven by genetic mutation and natural selection, slow, blind, cumulative. But now, there was a new force in play: culture. Ideas could spread horizontally, from mind to mind, far faster than genes could spread from parent to offspring. A good technique for knapping flint or trapping fish didn't have to wait for reproduction to propagate. It could be taught, learned, modified, and passed on within a single generation. And crucially, cultural innovations could accumulate. Each generation inherited not just genes, but knowledge. And they could add to that knowledge before passing it along. This is what scientists sometimes call cumulative cultural evolution, and it's the thing that truly separates us from every other species that has ever lived. Other animals have culture. Chimps use tools, whales have dialects, crows solve problems. But none of them ratchet. None of them build on previous generations' innovations in the way that turns a sharpened stone into a space telescope over the course of a few 100,000 years. The agricultural revolution, beginning around 12,000 years ago in the Fertile Crescent, and independently in several other parts of the world, was the next great inflection point. People began domesticating plants and animals: wheat, barley, rice, maize, sheep, goats, cattle. It wasn't a sudden invention. It was a slow drift, probably beginning with people managing wild stands of grain, then gradually selecting for traits they preferred: bigger seeds, plants that didn't shatter and drop their seeds before harvest, animals that were docile enough to keep penned. Over centuries, this reshaped both the organisms and the humans who depended on them. Agriculture allowed permanent settlements, food surpluses, population growth, specialization of labor. It also brought new diseases, social hierarchies, warfare over territory, and environmental degradation. It was, in many ways, a Faustian bargain. But it was irreversible. Once populations grew large enough to depend on farming, there was no going back to foraging. And from agriculture came cities, writing, mathematics, metallurgy, engineering in the formal sense, all the things we think of as civilization. The first cities in Mesopotamia, around 5,000 to 6,000 years ago. The first writing systems, developed initially for accounting, keeping track of grain stores and trade goods. Mathematics, born from the need to survey fields and predict floods. Bronze, then iron, each new material opening possibilities that the previous one couldn't. And through all of this, the same brains, the same biological hardware that evolved on the African savanna to track game, read social cues, and find water. We didn't get smarter in the genetic sense. We got smarter in the cultural sense. We built tools for thinking, language, writing, number systems, libraries, computers, that extended our cognitive reach far beyond what any individual brain could achieve alone. And here you are, lying in the dark, the inheritor of all of it. Every cell in your body carries DNA that's been copied and edited and passed along in an unbroken chain stretching back nearly 4 billion years to those first tentative self-replicating molecules in some warm, mineral-rich pool on a young Earth. You share genes with the cyanobacteria that first flooded the atmosphere with oxygen. You carry mitochondria that were once free-living bacteria, absorbed into a partnership so ancient that neither party could survive alone anymore. The calcium in your bones was forged in dying stars. The iron in your blood, the same element that colors ochre red, the pigment our ancestors ground and painted with 100,000 years ago. That iron was made in supernovae billions of years before the Earth existed. Your body is a museum of evolutionary history. The way your spine curves, the remnants of gill arches repurposed into your jaw and inner ear bones, the hiccup reflex that traces back to amphibian ancestors gulping air and water. All of it is a record, written in flesh and bone, of every adaptation, every compromise, every lucky break across an incomprehensible span of time. You breathe with lungs that evolved from swim bladders. You see with eyes whose basic architecture was laid down in the Cambrian. You think with a brain that expanded in fits and starts over 2 million years, fueled by cooked food and social complexity and the relentless pressure to understand the minds of others. And the story isn't over. Evolution hasn't stopped. It's still working on us, still shaping allele frequencies in human populations, still responding to the pressures of disease and diet and environment. But now, it shares the stage with something faster and stranger, our own cultural and technological evolution, which reshapes the world at a pace that biological evolution can't match. We've become a geological force, altering the climate, the chemistry of the oceans, the composition of ecosystems worldwide. For better and worse, the story of life on Earth is now, to an unprecedented degree, the story of what we choose to do. But that's a question for waking hours. Right now, in this quiet moment, it's enough just to know where you came from, to feel the weight of that long, long history, not as a burden, but as a kind of companionship. You're not alone in the dark. You're connected to every living thing that has ever existed on this planet, by chemistry, by ancestry, by the shared fact of being alive in a universe that didn't have to allow life at all, but did. 4 billion years of trying, failing, adapting, and persisting brought you to this exact moment, breathing in and out, your body warm, your mind drifting. And that's a remarkable thing, maybe the most remarkable thing there is. So let that thought settle. Let it be the last thing you hold before sleep takes you, that you are the universe's way of knowing itself, a brief flicker of awareness in an ancient, ongoing story, and that the story will still be unfolding when morning comes.